All right, let's begin. Welcome to the 812th monthly meeting of EPAASM. This is our Norman Willett Memorial Lecture. And we're very lucky to have Dr. Lori here from Davidson College today. And first of all, my name is Vincent Tam. I'm the branch president. We are here to encourage you to join National ASM membership to nurture this community of microbiology. And our national meeting is coming up soon, June 13th and 17th in Atlanta. And I just want to take this opportunity to express my gratitude to the Education Committee. We have a last minute conversion to the virtual format, and the Education Committee has really worked hard to make this happen. And while it's sad that we can't get together because it's so nice outside, we're really happy that everyone is able to join us today. And so we encourage you to join our branch. We had just last week, we had Monday, we had our student-run meeting, and they also run an annual Philadelphia Infection Immunity Forum. And of course, we have our annual symposium for clinical microbiology as well, and we have these monthly meetings. Our education committee, just want to give them a shout out of all the work they do. They have outreach program. They organize 2 career panels for our graduate students each year. They just recently did a lot of the science fair poster judging, and also they're working to go to elementary school where they talk to kids about the microbiota and making yogurts as a activity. And they're also working on a revision of the lab manual as a teaching tool. And so please go to epaasm.org. You can join the membership. For student, it's free if you just join a branch. And for faculty and postdoc, the branch membership is quite affordable. So without further ado, Bobby, the chair of the Education Committee, is going to introduce the speaker. And then for the questions at the end, you can either unmute yourself or we can type in the chat box and then we can read out the questions. So thank you very much.
Thank you, Vincent. Well, I wanted to say good evening and welcome to the 2024 Norman Willett Memorial Lecture. My name is Bobby McHale, as Vincent said, and I'm the co-chair, along with Brian Forster of St. Joseph's University, of the Education Committee of the Eastern Pennsylvania branch of the American Society for Microbiology. We want to thank you for your flexibility in attending this meeting via Zoom. The change due to the presence of protesters on the Drexel University campus. The good news is that we can all participate in the program. The bad news is that we missed the delicious reception that was planned, and obviously our outside time. And I was just taking a look at the news while I was waiting, and it appears that the protests are still going on. So I think it's a good thing that we are on Zoom. I wanted to give you a bit of background that it's been a long-standing tradition of the branch that one meeting each year would be sponsored by the Education Committee and focus on the many different aspects of microbiology education. From those educating, from those educating high school students through postgraduate education. The concept was originally suggested many years ago by Dr. Norman Willett, co-chair of the Education Committee at that time. Dr. Willett was a professor emeritus of the Temple University Dental School. He was president of the branch from 1975 to 77, and a strong advocate for involving students in science education from kindergarten through high school. To this end, Dr. Willett developed science modules that were shared in a variety of programs, and students and teachers could take these back to their schools. In order to facilitate these programs, Dr. Willett was the recipient of many grants, including Howard Hughes Medical Grants and National Institutes of Health grants. Dr. Willett passed away in 2014. It doesn't seem like it's been 10 years, but his legacy lives on through these annual presentations in his honor. So today, also, as Vincent mentioned, we're pleased to have Dr. Willett's children here with us, Susan and Dave. So thank you for taking the time for that. So each year, the Education Committee invites a renowned speaker in the field of microbiology education to share his or her views with branch members and guests. This evening, we're delighted to have Lori Hayer as our speaker. Dr. Hayer, by education, is a mathematician. Dr. Hayer received her PhD degree from the University of Colorado, where she focused on applied mathematics, and her undergraduate master's degree in mathematics from the University of Texas. As it says here, Dr. Hayer is currently the Associate Dean for Data and Computing, is a professor of mathematics and computer science, and coordinator of the Data Science Interdisciplinary Minor Program at Davidson College in North Carolina. So the question is, why would a society focused on microbiology invite a mathematician to address members and guests? So it came to the committee's attention that Dr. Hayer was co-author of a textbook entitled Integrating Concepts in Biology, which revolutionizes how biology is taught at both the introductory college level as well as at the high school level. This textbook has been shown to improve student engagement, increase retention, and develop critical science skills, among other benefits. In addition to this textbook, Dr. Hayer has authored a number of essays and book chapters, and one that caught my eye, is Integrating Synthetic Biology into the Microbiology Curriculum. She's given presentations on a wide variety of topics, including microassays, bioinformatics and sequence analysis, RNA sequence folding and alignment, and gene expression cluster analysis, just to name a few. Dr. Hayer has been the recipient of many grants, including National Science Foundation grants and Howard Hughes Medical Institute grants, just like Dr. Willett was, and on a variety of topics. Dr. Hayer has received numerous teaching awards, including most recently the Engaged Scholarship Award and the Hunter Hamilton Love of Teaching Award. So today, Dr. Hayer will give us insights about bacterial computers, teaching an old microbe new tricks. So please join me in welcoming Lori Hayer.
Thank you very much. I think I've got my screen showing and myself unmuted, so I can see my screen, so we can go. Thank you for that warm introduction, very generous words, and for your generous invitation to give this lecture. I'm really very honored. I think it's a wonderful thing that you all do to remember a colleague this way and to be a part of that process, and honoring someone in that way is really meaningful to me. So thank you very much for the invitation. I do want to address the point that Bobby made about, why invite a mathematician to give this talk? I appreciate what you had to say about it. And I would say that I've given a lot of talks to biologists, and I love speaking to a variety of biologists. It's really what my career has been all about in communicating across and among many disciplines. So this is a wonderful opportunity to do that. It's always a little intimidating. So I really appreciate the generosity that you will show to me as I speak about a topic or a set of topics that has, been my life over the last 20 some odd years. And this bacterial computing really for about a decade of that time. And I hope that you find it interesting, inspiring, and maybe you'll learn some new tricks in the same way that we teach our microbes some new tricks. So okay, yes, I can go. I want to start with a brief overview of this subfield called synthetic biology. And that's where my work has been focused, again, for, in synthetic biology, about a decade. The idea of synthetic biology is sort of like genetic engineering on steroids. It's a very intentional way of applying mathematical modeling, computation, engineering principles, the way that engineers think about designing systems to biology. And it has a hierarchy of organization starting at parts, which you might think of as a gene, and building up to devices, which might be a few genes or a gene and a promoter, some kind of combination. It's really an abstraction of how you think about a device. And then on to the highest level of abstraction being a system that's supposed to accomplish something. And synthetic biology has many applications in various areas, and many of these are of great importance. And, it would take, I guess, a whole semester to really give a sense of all the different ways that synthetic biology has been applied in these main areas. And I'm really just going to focus on that one of technology, and that's been the focus of our work. So I will focus there. But the guiding principles are, I've already mentioned mathematical modeling and to an extent modularity and abstraction, and sort of the way we think about building biological parts and building them up into devices and then systems. But I would say standardization is one of the key elements that makes it all work. It's the magic sauce. So standardization is the reason there's a bunch of screws and nuts and bolts there, is because we know historically that was a really key development in the history of the world, to decide that there should be a system for how we build these parts, screws, threads in particular, how they should be designed so that they are interchangeable and that people can use each other's parts. You specify what system you use and someone else knows what that means, and they have parts that fit it. And that is really key to how synthetic biology has developed as a field. Another key aspect of synthetic biology is the community that is built up around it. And we have participated in iGEM a number of times. Our first trip was in 2006, and that was the second iGEM ever. It has been going for 19 years now, and I haven't been in a while now. But there are hundreds of teams now, I guess, per year, hundreds per year that are participating in, it used to be called a jamboree, and I really like that term for it because it is focused on undergraduate research. It is based on teamwork. There are high school students participating in this as well. So it's a broadly accessible competition. There definitely is a competition element to it, but at least in the years that I went to iGEM, it was a celebration of the work that the students had done and the amazing things that they accomplished, typically in a very short amount of time. Students and their mentors would form teams in the spring, as many of us do, to have students doing research during the summer. We'll recruit them during the spring and then get them up to speed and do the work in intense ways during the summer. And then sometimes the fall, you'll get a student, or two to follow up and do an independent study or maybe an honors thesis and wrap things up. And that's really how the iGEM timeline works. It is, above all, a community and a set of standards that people follow that builds this community. You can do synthetic biology without iGEM, and in fact, we did for many years, sort of graduated from iGEM. I'm not sure that's quite the right term, but we went on and published in journals rather than going to iGEM. So we just sort of changed our focus and our goals. But iGEM also has grown in those years since. So I think it's an amazing opportunity to get involved in this field for all those educators out there who are thinking, well, maybe I would be interested in synthetic biology. This is a place to go learn about it, connect with others doing it, whether you go to the jamboree or the competition, as they call it, or not. And just to give you a sense of what that competition looks like, they have tracks now in all these different areas. And every one of them is based on designing, building, testing organisms. So genetically engineering, typically E. coli. There are some that work with yeast or even human cell lines, but almost all of the teams will work in E. coli and build a system that attempts to, or at least is designed to, diagnose an illness, treat an illness, produce drugs, produce other objects or things. Just an amazing array of things you can do with synthetic biology. And you'll see in that bottom right corner, the high school track is called out separately. So the high school students are working in all these different areas, but they pull them aside as their own competition within iGEM. So lots of opportunities to get involved. What did we do? I want to give you a sense of, the details of a couple of projects, not every detail, but a sense for what those problems are like. And then a little bit about how we take that into the classroom, or how you might take it into the classroom or the lab, research labs, et cetera, to do synthetic biology with students. I think of research and teaching as almost one thing. So I'm talking about our research, but at the same time, this is our educational program. And at the end, I can break out some of the more specific educational opportunities that we've offered. So this was our team in that 2006 iGEM Jamboree, and it is an interdisciplinary team, both mathematicians, I'm a part-time computer scientist, and biologists, mostly undergraduates that you see in this picture, with 5 mentors, and from 2 different institutions. So this was a very challenging thing to work across the, with collaborators across the country, but also extremely rewarding. And again, I could give a whole talk on how we made that work and what was really beneficial about it. But let me go ahead and talk about the problem that we worked on, that year called the pancake problem. Yummy looking pancakes. I guess this is dinner time and we're missing out on that delicious dinner. So we'll just have to look at pancakes and wish that we were eating chicken or something that you were going to serve. But the problem is this. So this is a mathematical problem, but it's sort of a practical problem. Imagine that you're a waiter, going out to a table with a stack of pancakes, and the chef is a little sloppy. So they're all different sizes, and you would really like this to look like a nice stack of pancakes, and you would like to sort them with the biggest one on the bottom and the smallest one on the top so that you have a nice pyramid of pancakes. And carrying the plate, you might also be carrying a spatula that you can stick into the stack of pancakes at some point and flip them over and repeat until you have them sorted. So the mathematical question is, how many flips does it take? What's the minimum number of flips that you could apply to the stack of pancakes to get them sorted? Well, that's one version of the problem. Another version, and this actually was published in the American Mathematical Monthly under a pseudonym of Harried Waiter. Jacob Goodman was the mathematician and was sort of embarrassed to put this out there thinking that it might be trivial. And it turned out it set off a whole, decades of research on the problem. Another version of the problem that I will talk more about is two variations. One is not only was the chef sloppy in how big the pancakes were, the chef burned these pancakes on one side. So I would like to get them sorted from, largest on the bottom, smallest on the top, and golden side up. So when I put the plate down, everyone thinks they're beautiful. Also, I'm going to think about a two spatula version. So the stack is sitting on the table, and I can put one spatula in and lift some pancakes and then flip some over. Or another way to think about that is your pancakes are floating in the air, and you could just go in anywhere and grab a substack and flip them over. Either one does the same thing. So you could, a mathematician would call that a reversal, as opposed to the first problem, which would be called a prefix reversal. And you'll get to see the sort of math model of this in just a minute. This one had a famous paper associated with it as well. This is Bill Gates, that is the William Gates, only academic paper. So he published on the burnt pancake problem in the Discrete Mathematics Journal. But for the biologists out there, you may recognize this problem. This, in fact, is a genome rearrangement problem. I could compare the genes on the mouse X chromosome, for example, with those on the human X chromosome. I think I lost my text. There we go. And I find many homologs, so that many of the genes are the same on both mouse and human, but they're in a different order and orientation. And we have some explanations in terms of how this might have evolved, right? The copying mechanism just took a wrong turn. The DNA got folded on itself and the mechanism went the wrong way, and so a section got inverted. But that inversion or reversal is exactly what we saw in the stack of pancakes. So again, we ask the question, how many inversions or reversals does it take to turn a mouse X chromosome into a human X chromosome? Which we think, happened. that there were these evolutionary processes that led us from one to the other. And so having this mathematical measure of how many reversals it would take is akin to sort of a proxy for a measure of evolutionary time. How far apart are these organisms? And of course, you could apply this on various chromosomes and to different organisms, and there's some complications here, but it's at least a model for evolutionary distance between organisms. Let me show you a little mathematical model just to sort of fix the idea of the pancake problem. So here the pancakes are not so golden. Got some colors so that we can distinguish them more easily. And also using integers to represent the pancakes by size. So 1 is the smallest pancake, 4 is the largest pancake, and they are in order from top to bottom. They're the second biggest, the fourth biggest, the first, no, the smallest, and then the second. So the third, let me try that again. So the sizes are 1, 2, 3, 4, smallest to largest. So we see 2 on the top, and then 4, and then 1, and then 3. But there's a little sort of hashing of these colors where the pancake is upside down. So those are burnt side up, and I'll indicate that in this mathematical model with the negative sign. So this is my stack of burnt pancakes in some random order, and I want to get them in the order 1, 2, 3, 4. Now, I can only see a few faces, so I don't know how many of you have already solved this problem. How many reversals? Amy's got it. I can see Amy as she raised her hand. How many reversals is it going to take to sort this stack into the order 1, 2, 3, 4? And remember, we want them all positive numbers. And when I do a reversal, the signs will flip. So let me take the first stab at this. This actually is a prefix reversal, but I'm going to allow reversals of any kind. So I'm going to flip those top 3 pancakes. And this is what I get. I have mathematically turned this into 1, now -4, 2, and then the 3 didn't move. I didn't put my spatula down there. Okay, so you get the idea? Now, what's my next move? Be careful, because if you make the wrong move, you can go backwards, right? You can make this take much longer. So you might be already getting the sense that it's not obvious what's the best next move to make. I can't do it in 1 flip from where I am right now. It's going to take at least 2. Can I do it in 2? Well, let's try flipping these 3. If I take the bottom 3, so I lifted the first one and flipped the bottom 3. Now I have this arrangement. Mathematically, that is 1, -3, -2, 4. And now maybe you do see from here, I can make it in 1 flip because I have that. I can go in anywhere in reverse. I can reverse -3, -2 to become 2, 3. Voila, I have sorted this stack of pancakes in 3 reversals or 2 spatula flips. Now let's think about what this looks like in an organism. So what are DNA pancakes? Well, the pancakes themselves are just sequences of DNA, but I may want to think about them being some functional pieces of DNA, whether they're genes or promoters or ribosomal binding sites or some kind of functional element or parts of those things actually might be a good idea, and we'll talk about why. I'm going to start with some just abstraction for those genetic elements and call them still 1, 2, 3, and 4. And imagine that I have them in this sort of arbitrary order to begin with. And I would like to do the inversion process biologically, get these sorted. Oh, it looks like it's not going to take much. I kind of went into the middle of the history there and started 1 flip away from the end. All I need to do is flip those middle 2, and I'm going to have this thing sorted. Well, what do I need? I need a flipper. I need a spatula. So there is such a thing. Hin recombinase comes from Salmonella. So we borrow that and put it into our E. coli and say, okay, do your thing. What Hin does is recognize these sites that we have strategically placed in between our genetic elements, and it acts like scissors. It will cut in those locations, and the sites are palindromic. So it might go right back in the way it was, but it might also flip over and reinsert flipped. And in Salmonella, this has a function, right, of, I may have forgotten. It changes something in how the Salmonella deals with its environment. So it can flip that switch and say, okay, now I need to act this way. But we borrowed it into E. coli and use it, engineer it to do this particular function for us. So it can take that element and flip it over into the sorted order. So the way we might think about this as a computer, a living computer, in fact, is to take this whole cassette, this arrangement, and put it in the E. coli living cell with the Hin recombinase, and then just turn it loose and use time as a way to count flips. Again, that's a proxy, and we need some mathematical modeling, and we're going to have to, let some time go by and then see what's in the population and see again and again and again to get a good reporting system to tell us when this thing is all sorted. We're going to have to think carefully about what elements are in these pancakes. Maybe we could design this with 4 different parts of GFP, and only when they're in the sorted order would this thing glow green. So that's an idea. It's an idea that we used, but it was a little bit complicated because you have to have these Hicks recognition sites for the Hin recombinase that split up your gene elements, right? The gene itself, if that's what you're doing, splitting a GFP into 4 parts, you're sticking stuff in there that it's not going to be happy with, right? It's going to maybe not still form its shape and glow green. So we were able to split GFP into 2 parts, but splitting it into 4, we didn't get. So we used other elements, and just, if one of these is a promoter and one of it is an RBS and the other 2 are GFP, then you could get all 4 of those in order and know that you had them in order. Of course, you could always sequence and find out if they're in order. So there's that, right? But the whole point of these bacterial computers, and maybe just take a step back from this particular example, why would we ever want to do such a thing, right? We have computers. I've got one right here in front of me. Why would we want to make E. coli solve mathematical or computational problems? Well, one reason is literally these living computers. It could be any kind of organism or many kinds of organisms that we put them into. And then you could imagine them performing functions in environments that real computers can't go. So the depths of the ocean, outer space, inside of our bodies. We could have these functional computers solving maybe simple problems, although this is mathematically a very complex problem that I've presented. It's, NP complete. It's a hard problem. But if we get this sort of proof of principle that we can compute, we can solve problems like this, you could imagine applying and doing computing in these almost anywhere, right? Anywhere that living cells can go. A couple of other bacterial computing applications that we did, I'll just mention briefly. This was the following year, we actually built on the idea of pancakes to solve the Hamiltonian path problem, which is another famous mathematical, hard computational problem. And I don't have time to go into all the details, but the idea was this gene splitting idea to represent the edges in this network, the lines between the nodes in the network. We would represent those edges as a pancake, but then a pancake would be two half genes, again, requiring us to split genes. And we ran into challenges with that, but it's certainly not impossible. And you could imagine a system for finding what genes can be split this way and having lots of nodes in your network and solving this problem. This one actually got, we got the best paper of the year in the Journal of Biological Engineering for both of these projects. This one actually got a lot of popular press as well, because the Hamiltonian path problem is so well known, or people just got excited about it. It's, I don't know, they don't like pancakes, but they like networks. Anyway, you can go back in history and read some of those stories. We tried a cryptographic hash function using E. coli, and we attempted to solve a satisfiability problem. Again, for computer scientists, this is one of the most fundamental problems that they face. All problems that are hard can be turned into a satisfiability problem. So that's of a lot of interest computationally. And as you can see, with our undergraduate teams, we had a lot of fun coming up with team names and logos and t-shirts and, the picture that I showed earlier, that's us at MIT, really just having a ball with science and other scientists, learning from each other and all of them. The last time we went to iGEM actually was the knapsack problem, another sort of hard mathematical problem. But then we took a step back and said, what's really the most important problem to solve? One of the hardest computational problems that we have tried to solve is trying to model and optimize gene expression. So what if we could program the cells to solve that problem? And that is a project that we worked on for several years and completed. We did get this one published a few years ago now, called Programmed Evolution. So we know that if we try to, say, produce drugs, have E. coli produce some useful compound, we want to make them produce as much as possible. Just turn everything up all the way to 11 and make it spit stuff out as fast as it can. That's the whole point. That's how we can be successful in producing a compound. Well, let's look inside one of these cells. How do we do that? We have some product that we're trying to make. In our case, we were going to produce theophylline. It's an asthma medication. And it happens to very conveniently be very close to a compound that is readily available, and that is caffeine. And there is a gene, caffeine demethylase. You can see these are only different by a methyl group. Caffeine demethylase will convert caffeine into theophylline. So if we put this gene into E. coli and give it caffeine, voila, it will make our asthma medication and we'll be rich. Well, why is this a hard problem? Right? We know that one of the hard problems is that cell that we engineered is going to be under some pressure. There's all these other cells out there that don't have to carry around all that baggage, and they are cheap, relatively. They will dominate the population. So that we carefully engineered this thing, but it will evolve out of that configuration into one that it prefers and does not any longer make our asthma medication. So natural selection is the enemy here. So to do synthetic selection, we want to reward the cell that we engineered and punish and remove, that's the Wile E. Coyote treatment, remove these cells that we don't want around. So how might we do that? So it's like engineering on top of the engineering. We need to engineer these parts that will reward and punish the things that we want to reward and punish. There we go. We're going to have lots of successful cells, and minimize these ones that are not doing what we want. Just knock them out. So how do we do this engineering job? Our idea, which is in this PLOS 1 paper with 49 undergraduate students, right? So again, this took a few years across 2 campuses. We worked with a lot of students to get this done. I'll give you sort of the big picture, and then I'll kind of speed through the details to give you a sense of how we did this, but maybe not in as much detail as you want. I can answer questions later. But the idea is, we don't know actually that a combination of a high copy number plasmid and a really strong promoter and a really strong RBS and a particular allele of this caffeine demethylase that seems to be, highly produced. We don't know for a fact that those are actually optimal. There may be things that we don't understand about how this system works. And that's the whole point of programmed evolution, is in letting the bacteria solve this problem for us. Like mathematicians like me, when we try to model something like this, first question we ask is, well, what are the values of all the parameters? What are the rates of this, that, and the other? And no one knows, right? So you can't actually build these models and effectively predict what's going to come out, because we only have guesses at most of these parameters. And then when you put in new things, they don't work the way that they did when those things weren't there. So really, it is just guessing. But the cells know, right? They have a hard time telling us, but they know. So the system is designed for them to tell us the solution. So first, we need some kind of combinatorics module. We need to be able to produce a lot of different genetic variations that we want it to choose from. Then we need the anvil. I guess it is partly the money bag and partly the anvil. We need some kind of fitness module to say, only if you retain this will you, the system that we're trying to get it to do, like only if you retain this converting from caffeine to theophylline function, only then will we let you live. And, or at least we're going to punish, severely those that are not producing. And then we would also like to have a glimpse into what's going on here. So we're going to build a biosensor module to sort of report to us the status of this system. And then repeat this cycle to get evolution to happen, right, in this sort of forced way, and tell us the solution in the end. Well, the combinatorics module, we used a technique called junction Golden Gate assembly, and I can show you Golden Gate in a little bit, but it's a way of quickly putting together lots of biological parts with this system that makes them fit together. Okay. Designing those junctions actually is an interesting computational problem, trying to get them to be compatible and, operate at the similar melting temperatures and all kinds of things that we had to think about. But the idea is just make lots of versions of this that are modularly put together, right? So that these different versions of the promoter, different versions of the RBS, and different versions of the gene can be chosen and fit into to make these different, it's a combinatorial explosion of these different constructs. Gadget is the tool that my students built, and this is something that happened every summer in synthetic biology. Is there were things that we were trying to do biologically that were either very tedious, very easy to make mistakes on, usually both, and we needed some kind of a tool, an online tool, some kind of program that math and computer science students could build for us to make this job easier. So in this case, choosing the sequences for those junctions and then automatically telling you what primers to use so that you can construct these things and you don't make mistakes in all those steps. Here, there's actually some more sophisticated computational stuff going on to choose a set that have a similar melting temperature. So it's doing a lot of things there. The biosensor module, and then this is going to feed into the fitness module as well. We used this idea of a riboswitch. So if you have theophylline and this riboswitch, it's one that was already known in the literature. Theophylline is cognate aptamer here in this DNA. Oops, wrong way. Yes, okay, some clever animations, boom. It will change the conformation of this RNA and reveal or, uncover the RBS so that the gene can be expressed. So in the presence of theophylline, this riboswitch will cause this gene to turn on and you will see green. So seeing green is one thing, right? And we did. And so we sort of proved that that worked. But you can also use this same idea for your reward and punishment system, your fitness module. If you have theophylline and you turn on this riboswitch, you could also grant the cell some antibiotic resistance. So just by putting a different gene downstream of the same riboswitch, we sort of get two for the price of one. This one function of recognizing theophylline and uncovering an RBS gives us everything we need to reward the cells and know that it's working at the same time. This is a chart of our results, and I guess the main point I wanted to drive home here, not try to explain it all, is simply that the optimal performing constructs are not the ones that we might have picked as humans. For example, they were on low copy number plasmids. And, a high promoter does well, but a low RBS does better. Those kinds of things we would not have predicted, we can see in these results. We saw, I haven't shown these results, but we saw evolution happen. We did a population sequencing study to see what is here after some time has gone by. And although we started with equal percentage of each one of these constructs, we ended with very unequal percentages. So we forced evolution to happen, and we got to emerge as the dominant population, those that were the most fit. And they were doing the job that we wanted them to do. So we kept the function, and we had, cells that continued and did not evolve out of that desired solution. Just checking the time, and I know we want to leave some time at the end for questions. So I'll again sort of quickly go through this section, but I just wanted to give you a sense of what are these standardized parts and how familiar might they look to you. So the original system of biobricks that iGEM folks created were just based on these sites that you could cut, these restriction sites that you could cut in a clever way, and they leave scars behind, but then they're not cuttable again. So if you look at the sequences of all of these things, you can see how this works, that if you have the blue part in one plasmid and the green part in another plasmid, they both are constructed the same way with EcoR1 and XBA upstream and SPE and PST1 downstream. And you cut the first one with EcoR1 and SPE1, you cut the second one with EcoR1 and XBA, and then they're compatible with each other. So when you mix them and ligate, the blue part pops in front of the green part. Classic techniques. The key here is, like, everybody agreed that this is the system we're going to use for all of our biological parts in iGEM and more broadly in the field of synthetic biology. But this was the sort of version 1. Version 2 gets away from some of the problems that you have with that system of background and so forth. Just it pumps up the number of antibiotics that you have to have, the number of different kinds of plasmids that you have to have. You have one that's sort of your receiving plasmid. The other two you cut similarly as we saw before, and then they both pop into the receiving plasmid. But what we use more so, and many people do because it's very undergraduate friendly, very fast, easy, cheap, is Golden Gate assembly using type 2S enzymes. BSA1 is what I'm showing here. Importantly, this site is not a palindrome. And the cut is either up or downstream from the recognition site. So this is like no restriction enzyme that most people have not worked with these, right? It's like, what is this system? Well, the system is really powerful. So you cut upstream or down, and I want to get to, well, okay, I'm going to show you in the next section, actually, a plasmid that has this in it and how we use it. So that falls into this teaching synthetic biology or teaching with synthetic biology. Again, in my view, in my experience, those are kind of one and the same thing. We teach all these fundamental biological ideas and techniques with synthetic biology. So you're teaching synthetic biology, but kind of you're teaching all of these molecular and genetic techniques. Okay, here's the part. So this is, that's in the catalog. That's the number that it has in the iGEM catalog of parts. So you can go, find a sequence and get it for yourself. And in fact, I'll show in a minute the link that this is available as a kit. So you can just get this thing if you want to not build it yourself. Or if you're an iGEM, they might just give it to you. But it has GFP on the left, pointing to the left, RFP on the right, pointing to the right, and it starts out with a promoter pointing to the left. So it's going to express GFP. If you put this in a cell, it's going to glow green. But we've got these BSA1 sites on either side of the promoter. So we can cut in those two locations, as I showed those sort of upstream and downstream cuts. And pop that out, it might pop right back in. Or if we provide an alternative with the same sticky ends, then that one might pop in instead. So that's the goal. And we use this in our first bio course, Bio 113, lab section, as several weeks of the lab. The students, learning these techniques and picking a promoter that they want to test, and then using this system to clone that promoter in and then see how it functions. So they start with this plasmid and the promoter of interest. They get it into the plasmid and then they test it. We call this plasmid p-clone red. If they only had the original, they didn't get any ligations of their promoter of interest into the plasmid, then it's going to, every colony is going to be green. But if they got some successful ligations, there usually is a mixed population. You're going to see some where it just popped right back in and still glows green. The original promoter was reabsorbed. But the ones that have the new promoter are red, and you pick those colonies and go. So, if it's not there, you ignore it, and if it is, you can use it. So this is available, as I said, from Carolina Biological as an entire kit, but we don't use the kit, right? This is not a really difficult or really expensive thing to do, and students learn lots of things along the way. Also, in the teaching tools, I wanted to mention, Bobby talks about our textbook, and that's a way that you can teach synthetic biology as well as in the lab. We don't have a big focus on it in our book, but we do have a couple of sections where this appears. This is one of them. It doesn't sound like it's about synthetic biology, so you have to go read it to see what all this is about. But it's got some of these switches and sort of understanding gene expression. Another important aspect of teaching synthetic biology or teaching with synthetic biology is the incredible breadth and depth of ethical implications that it raises. And students love discussing this. That was an important consideration in our textbook, was to include these discussions and discussion questions to help our students grapple with these important questions. And synthetic biology definitely is an area where there's a lot of them, but we've got these, throughout the textbook. And then I want to talk about our group, or our little band of collaborators and a broader community called GCAT, which is still sort of out there somewhere. It's less organized than it once was. But this is Jeff Poet on the left from Missouri Western, Todd Eckdell on the right from Missouri Western, and then Malcolm Kimball, my colleague here, and myself acting out GCAT. But the four of us this was also at MIT, the four of us have led workshops for faculty and sort of spread this technique. Maybe some of you and many folks out there have participated in these workshops. This is actually the very first workshop we did, which was on microarrays, and the very last workshop we did, which was synthetic biology. But that, teaching the teachers, I think, is a really important way to make educational progress, right? To spread ideas, to give people confidence. I was talking to Bettina earlier about these and how important it was that we had pairs of faculty. There would be a biologically oriented faculty member and then a more quantitatively oriented faculty member come together. You had to come with a partner so that when you left, you had that collaboration ready to get off the ground. And many of them have been very, very successful. So, can you do it? Well, yes, I haven't shown proof of all of these claims, but the expenses are relatively low. We work with rising sophomores in our lab all the time. So once a student has taken sort of intro bio course, they're ready to do this work. And they're ready to be, they love the creative aspect of this work, right? To imagine something that you might be able to do with a synthetic system and then try to find the genes that would do it and put them together in a system that would actually work is incredible fun. And because of the types of things that you have to do, it's great fertile ground for a collaboration with a mathematician or a computer scientist. There's lots of modeling and just these design questions actually are things that they enjoy, and statisticians come in too. So there's plenty to go around. And just circling back to that, can you do synthetic biology? Is this something that you want to take to your classroom or your lab? There is this huge community out there to join and be a part of and contribute to, and have them contribute to you. So just to close with thanks for all the people that I've worked with. I couldn't fit all the student names on here, but I'm so grateful for their participation, for their sort of being willing to take this risk and do something that sounded impossible and getting it done, and also really grateful for the funding that we've had over the years. So I will stop, and I hope there's still some time for questions.
So if you want to type your question into the chat, that would be fine, or you can address them directly to Dr. Heyer. So I have a question for you, Laurie. Is there any age group that you found that it just doesn't work well with? They don't have the sophistication to understand what's happening?
Well, I personally have not worked with high school students, but I know many have, right? It's a whole section at iGEM, so I think high school students are not too young to do this work. Again, there's some basic techniques that you need to build the systems, but they're accessible, they're safe. We've put protocols online, and many others have, right? It's a very open community, so it's very easy to find the protocols. We've built some sort of paper activities and things that you can take to students who may be a little less comfortable and need to play with it a little before they're ready to go into lab and do it for real. So I don't think at some point there's a lower bound because they can't pipette, or they can't do some of the lab work, but I think it's very intellectually, this is very approachable. You just might scale the type of problem that you're going to do, right? You don't start with the Hamiltonian path problem, right? You start with characterizing promoters in p-clone red, sort of that last example that I gave. That one's not only very accessible, but it helps drive home the lessons that you're trying to teach anyway about these genetic elements and then how does gene expression work, and forward and back, different strands of DNA. And so many ideas are captured by that one construct and that one experiment that they're doing with it. And then doing the research on promoters And oh, some promoters are temperature sensitive and some are, sensitive to other chemicals that I'd be able to put in here and test, and they start making hypotheses. So it's an incredible journey for those beginning learners. I think to get up to the complexity of bacterial computers, yeah, then maybe you're talking like we worked with rising sophomores and not only did those, but it like decided what we were going to do and invented those systems with our students. We didn't do it for them. They really drove the research.
Thank you. So Amy Vollmer has a question. She wants to know who has the library of parts for synthetic biology. Years ago, we received them from a workshop held at HHMI's Janella Farms.
Yes, a wonderful time we had there.
Oh, that was great.
So the iGEM presumably still has a library that you get if you pay to join, the competition. We have had a library, that we sent out to people of, a much smaller set that we knew worked, that we shared with folks. So finding the sequences for all of those parts is easy to do, and we still host to that. Getting the DNA sent to you is a different thing. So either you're getting into iGEM and you're paying for it, or you're collaborating with somebody that has it in their freezer, which before Malcolm retired, I would have said, yeah, just email Malcolm. But he retired this year, and that's harder to do.
Is Todd still at
Todd is still
Western Missouri?
Yes, Todd is still working, so email Todd.
Todd Dahl is a fantastic person. If anybody wants to get in touch with him, he'd be glad to help you. Yeah. Oh, good. Because I know Malcolm retired. That's kind of why I asked. He's the person we got our strains from, but they got all used up. I mean, it was great for a while.
Yeah. Now we
So
need
another question from Joanne regarding the high school route. Does IGM function like a robotics competition where there is a yearly problem and a culminating expose, or is it more like a showcase where every student group displays their ideas that they choose to integrate the concepts?
Yeah, it's the latter. And in fact, at the jamboree or the events, they now call it a village. So they put, these groups together so that they can go around and see each other's posters and talk to each other by village. And that is the tracks that I showed earlier. So the high schoolers would all be, close together and, share each other's ideas, but they're not all working on the same challenge. They each have their own.
Okay. Thank you. Are there any other questions you'd like to ask Dr. Fire before we leave?
Yeah, Lori. Thanks for a great talk. So I'm just curious, you're talking about how you work with the rising sophomore, but I guess do students return year after year and maybe they can help out the younger students and that kind of collaborations and have more say in terms of how to solve problems or that kind of collaborations?
Yeah, so this evolved over time and how we approached it, but typically we would, in the iGEM years at least, we were starting a whole new problem every summer with a whole new team. And as you may be implying, I'm sure you can appreciate that's challenging. Untrained students, well, that's okay because that's part of the point, right? In fact, that's the whole point for us, is to train students. So starting with raw recruits, not really a problem, a challenge, but a good challenge. If you had what we later called finishers, the finishers then could help mentor those students. And I think if I had it to do all over again, I would start that from the outset, right? That there would be peer mentors who had been through this process. I've grown to understand and appreciate the value of peer mentors. It seems to become more and more valuable as I don't know if it's a culture shift or what, but I have seen that in math classes and computer science classes, and I definitely think it would be true in the lab. Peer mentoring would be really a big boost. But at the same time, you want that team to have their own project, right? You want them to have their own freedom. I think that's an important part of this field, is that design phase, that creativity. So the finishers for us, and this is how it worked sort of in the later years, was, we'd get us to a certain place by the iGEM competition, but not really done, right? Certainly not ready to publish. And so we would work with students we called finishers the following year. During the academic year, maybe as independent studies or honors theses or whatever mechanisms, some for pay, would come and finish up that project. And sometimes even bleed over into the following year. They would get their funding for research the next summer and finish a project that they had started the previous summer. So there's lots of different models, and I think the peer mentoring definitely is valuable, and it's very hard to get everything done, from idea, concept, all the way to publication in a summer. So having some way to come back to it and finish it is really important.
I just want to sneak in one science question. So really interested about your expression optimization, right? So when you did sequence the populations, do you find any point mutations that unexpected, for example, the RBS change that you wouldn't expect to have better expression, that kind of results?
So the answer is yes. I don't know that I can give a specific example, but I think yes, there were some unexpected mutation. You would expect actually there to be some evolution to do even better than what we gave them to start with. Yep.
Great, thanks.
Well, thank you very much, Dr. Hare, for coming to Zoom. Unfortunately, you couldn't come to Philadelphia, but maybe another time. But thank you for a very interesting and informative presentation. So
Thank you so much for having me.
thank you.
Thank you. Fantastic.
Thank you
So
very
much
much.
fun.
Thank you.
Take care, Lori.
Thank you, Susan.